If you like this video, please subscribe. Thank you.
Importantly, I want to just kind of remind you that what we were doing in the last lecture is talking about this distinct step, the pyruvate dehydrogenase complex. And also I want you to keep track that pyruvate most likely came from glucose. Okay, meaning you are in a well-fed state, there's plenty of glucose, you form pyruvate. Pyruvate dehydrogenase produces acetyl-CoA, and from two pyruvates, two NADHs, which is really an important energy output, significant energy output. Now, the fate of acetyl-CoA is shown here. There's a catabolic fate, which means you further oxidize the two carbons. These are two carbons. Acetyl has two carbons. CoA is the coenzyme. So these two carbons can be oxidized all the way to CO2. And as I said, for most cells, especially the aerobic cells and tissues, that's a very important fate. So the brain, for example, will take glucose, oxidize it to pyruvate, break it down through glycolysis to pyruvate, pyruvate to acetyl-CoA, then all the way to CO2. And the way we do that is through the Tca cycle. But I want to remind you of two other important things, that this acetyl-CoA can be used to make fatty acids, to make cholesterol. So it's an important intermediate in anabolic processes as well. So we'll talk about that, this part here in the third block, as well as ketone body synthesis. Also importantly, I want you to appreciate that acetyl-CoA can be derived from other molecules. Some amino acids produce acetyl-CoA as a keto acid, and the oxidation of fatty acids can produce acetyl-CoA. But for now, let's assume that acetyl-CoA came from pyruvate, from glycolysis, from glucose, and we'll go into the Tca cycle for further oxidation. So the Tca cycle has three names: tricarboxylic acid cycle, the citric acid cycle, citrate cycle, or the Krebs cycle. So they're all synonymous, referring to the same thing. Importantly, pay attention to the word cycle. A cycle is where you use up something in the beginning and you regenerate it at the end, versus a linear pathway where that doesn't happen. Okay? So we're gonna use up something in the beginning and regenerate what we started out with. So the term tricarboxylic acid is based on the first molecule made in that cycle, which is citric acid or citrate. That's a tricarboxylic acid. So that you get all that nomenclature down. So what I'll do here, I'll go through kind of the big picture overview and try to point out to you where you want to pay attention. Well, you really want to pay attention to everything, but there is specifically a few steps that are more important in a way. So first of all, you don't really need to know any of the structures. So don't worry about the structures. They're there to help you maybe visualize, you know, what's happening or what's being lost and so on. So let's start here, the beginning of the cycle. Again, remember, ask yourself, what is that cycle for? It's to produce energy from acetyl-CoA, oxidized acetyl-CoA. Also, ask yourself, where is it taking place? It's taking place exclusively in the mitochondrial matrix. Okay? So those are very important key things to always ask yourself about every pathway and cycle. So we're starting with acetyl-CoA that came from pyruvate through pyruvate dehydrogenase. You want to condense. This is a 2-carbon molecule with another molecule called oxaloacetate. We've already covered that in gluconeogenesis, but this is oxaloacetate that is going to condense with the 2 carbons there to form the first important intermediate in the Tca cycle, citric acid or citrate. So you add 2 plus 4, that gives you 6 carbons. That has 1, 2, 3 carboxylic groups. That's why the cycle is called the TCA, or the tricarboxylic acid cycle, or the citrate cycle. So the first molecule formed is citrate by enzyme citrate synthase. That's the beginning always of the cycle. The cycle will always begin here on top. So you need both acetyl-CoA and oxaloacetate to start it. Then you have this enzyme that will move around some of the molecules in citrate to form isocitrate. Okay, you didn't lose any carbons, they've just been rearranged. You can see where that the hydroxyl was shifted and so on, but it's the molecule that is produced is isocitrate. The enzyme's name is aconitase, so it's a type of isomerase. Okay, you're just moving things around, you're not losing any molecules. Isocitrate is now going to be oxidized. Okay, and oxidized again, if you lose a carbon in the form of CO2, that's an oxidation step. Okay, so the first enzyme here involved in that is called isocitrate dehydrogenase. Here's the CO2. You are going for, this is just an intermediate, don't worry too much about it, but you're losing a CO2, so you go from a 6-carbon tricarboxylic molecule to a 5-carbon dicarboxylic molecule. You lost one of the CO2, one of the carboxylic groups, this one in fact, and gives you alpha-ketoglutarate. Again, the best way to always think about oxidation reduction reactions is to remember, if you oxidize something, that means that molecule lost electrons. Those electrons are going to be gained by something else. They don't just disappear in thin air. So, and that something else therefore will get reduced. So you lost CO2, you oxidize isocitrate to alpha-ketoglutarate. The recipient of those electrons will be NAD plus to give you NADH. So NADH is a high energy molecules that has gained electrons. Okay? So that's obviously an important step because it's producing energy. You want to pay attention to it. We'll talk about this enzyme some more. But you produced, and you can think of this CO2 really is as you've already oxidized one of the carbons that you started out in acetyl-CoA. That's one way to think about it. So you have now alpha-ketoglutarate. Alpha-ketoglutarate is going to be also oxidized by
alpha-ketoglutarate dehydrogenase.
Lose a carboxylic group, and because of that oxidation, you're going to reduce NAD plus to NADH. So you produce another molecule of NADH. Simultaneously, as you oxidize that CO2, you add a CoA, the coenzyme A, to what's left of alpha-ketoglutarate to form Succinyl Coa. So we've gone from a 5-carbon to a 4-carbon molecule. And it's really important to pay attention there. This is a very important link bond, high energy bond, Succinyl Coa. This molecule now, because of that high energy bond, if you break that high energy bond in the following step by either Succinyl Coa synthase, the enzyme is called Succinyl Coa synthase or Succinate thiokinase. Same enzyme has two names, Succinyl Coa synthase or Succinate thiokinase. When you break this bond, you produce enough energy to synthesize, to make GDP plus an organic phosphate not shown to form GTP. And GTP has the same energy as ATP. So again, this is an energy producing step. So thus far, we produced 2 NADHs and a GTP. So when you form that, you produce succinate. Succinate gets oxidized to fumarate, and if you're not sure, there's a double bond here created in fumarate. And because you oxidized succinate, you're going to reduce something else, FAD, the coenzyme here, to FADH2, and that's another high energy molecule. So you produce another high energy molecule in the form of FADH2. Fumarate then is converted to malate by an enzyme called fumarase. It's just a hydration reaction, and then malate is re-oxidized or oxidized by malate dehydrogenase. That's the last step in the cycle if you want. Again, you oxidize malate back to oxaloacetate. You oxidize one thing, you reduce something else, so you form NADH. And that's the last high energy molecule formed by malate dehydrogenase. Importantly, you have regenerated oxaloacetate, what you started out with. So in essence, the cycle has been used to oxidize acetyl-CoA by producing 2 CO2s. Here's one, here's another one. And think of them as the carbons that originated in acetyl-CoA. So you have processed that acetyl-CoA through the Tca cycle. Once you regenerate oxaloacetate, the cycle can continue, meaning if you have another acetyl-CoA coming in, now the cycle can restart over and do the same thing again. So let's say that acetyl-CoA was coming from pyruvate, and that pyruvate was coming from glucose. So you're going to have 2 pyruvates, and you're going to have 2 acetyl-CoAs. And that means the cycle has to go around twice. Each time it will pick up 1 acetyl-CoA and produce these energy molecules, and then it can repeat itself for the second acetyl-CoA. You all with me? Kind of? Okay. So first of all, you want to pay attention to where energy is produced, where CO2 is produced, where NADH is produced, which enzymes produce those molecules, where GTP is produced. That's another energy producing step. where FADH2 is produced. So you have 3 steps that produce NADH, one that produces FADH2, and one that produces GTP. So you want to know the sequence, the name of the enzymes, but don't worry about the structures. So we'll go through each of these enzymes kind of quickly. Here's the first one, oxaloacetate acetyl-CoA to form citrate. Citrate then is isomerized by aconitase to form isocitrate. Isocitrate is oxidized by isocitrate dehydrogenase. So you're losing that CO2 and producing the first high energy molecule. I'll get to this here a little later, but I just want you just to know the sequence right now. Isocitrate to alpha ketoglutarate. You've lost 1 carbon in the form of CO2 and produce 1 NADH. Alpha ketoglutarate is then oxidized by alpha ketoglutarate dehydrogenase. Again, you lose another CO2, producing another NADH molecule, and simultaneously you're going to attach a CoA to the succinate. There's the CoA to form Succinyl Coa. Actually, this is a very important enzyme in the TCA cycle. And it is similar to pyruvate dehydrogenase in that it requires all the same coenzymes: NAD, FAD, CoA, lipoic acid, and thiamine pyrophosphate. Okay, so this has the same requirement of coenzymes as pyruvate dehydrogenase. So you form Succinyl Coa, and then you break that bond. I said this enzyme is that breaks that Succinyl Coa thioester bond. It's called succinate thiokinase or Succinyl Coa synthase. And that energy produced allows the synthesis of GTP from GDP and inorganic phosphate, and that's really equivalent to ATP. GTP or ATP are kind of about the same, have the same energy in them. You produce succinate. Succinate dehydrogenase produces another high energy bond when it's converted to fumarate in the form of FADH2, and then fumarate is then hydrated by fumarase to form malate, and finally malate, not shown here, I don't think I have a slide on that. Malate is re-oxidized or oxidized to oxaloacetate producing NADH, and importantly regenerating the oxaloacetate we started out with, and that and that allows the cycle to continue. So here's a simpler just summary of the all the enzymes: citrate synthase followed by aconitase followed by isocitrate dehydrogenase, again NADH, CO2 produced, 4-alpha ketoglutarate dehydrogenase, CO2, NADH produced. By this point, and Succinyl Coa, by this point, you've already lost the CO2s. Here they are. So you've kind of oxidized, think about it, you've oxidized the 2 carbons of acetyl-CoA. Succinyl-CoA thiokinase, succinate thiokinase or Succinyl-CoA synthase is the enzyme that breaks this bond to CoA, generating enough energy to synthesize GTP. Then step 6, succinate dehydrogenase produces FADH2 and fumarate to malate by fumarase and malate to oxaloacetate bimalate dehydrogenase to produce NADH. So pay attention to all of these orange molecules and purple, really this one is purple, but the 3 NADHs, 1 GTP, and 1 FADH2. So each time you oxidize an acetyl-CoA through the Tca cycle, you produce these high energy molecules. 3 NADHs, 1 FADH2, and 1 GTP. NADH, each NADH will have the equivalent energy of 3 ATPs. And we'll get to that on Monday next week, how we get to that. And each FADH2 has the equivalent energy of 2 ATPs, or is equivalent, able to produce the energy of 2 ATPs. So if you do the math, every time you oxidize an acetyl-CoA in the Tca cycle, you produce 3 NADH, 1 GTP, and 1 FADH2. So the total ATP output, 3 times 3 is 9. That's equivalent to ATP. 1 times 2 and 1. The total here is 12 ATPs from the oxidation of a single acetyl-CoA. Again, I want to keep you linked into, say, glycolysis. If you had 2 pyruvates that went through pyruvate dehydrogenase, then you would have 2 acetyl-CoAs to be oxidized. Then your output of energy would be twice that, would be 24 ATPs. The cycle will go twice, each time producing 12 ATPs. Okay? So you should just at first look realize, wow, that's a lot of energy. 24 ATPs from the Tca cycle going twice, oxidizing to acetyl-CoAs. That's actually 3 times as much energy as was produced from glycolysis. 24 ATPs, glycolysis a maximum of 8 ATPs. So
That's pretty impressive in terms of energy production. If you like this video, please subscribe. Thank you.